Okay, this is chapter 19. This is the heart lecture. Chapter 18, 19, and 20, those are our cardiovascular systems. 18 was the blood, this is the heart, and then 20 will be the vessels. Hold on, click back into it. There we go. The cardiovascular system is your heart and blood vessels. It transports blood throughout the body. It's delivering oxygen, nutrients, and removing carbon dioxide and wastes, providing adequate perfusion, which is the delivery of blood per time per gram of tissue. It's measured in milliliters per minute per gram. So delivery of blood per time per gram of tissue. Adequate perfusion is necessary for sufficient delivery to maintain cells' health. It requires continual pumping of the heart and open, healthy vessels. Our heart starts pumping and it doesn't stop until we die. Constantly doing this, delivering oxygen for metabolism, nutrients, glucose, ions, minerals to help with building cellular structures, and then removing our carbon dioxide and wastes. Your blood vessels, you have three main ones: arteries, veins, and capillaries. Arteries carry blood away from the heart. I always think arteries away, A and A. Most of them, but not all of them, not all of your arteries, carry oxygenated blood. We'll see it's the ones coming from the heart directly that do not carry that. The veins carry blood back to the heart. Most, not all, again, carry deoxygenated blood. We'll see that in the pulmonary circulation. For your capillaries, capillaries are sites of exchange of gases between blood and air in the lungs and between blood and body cells. We call this external respiration and internal respiration. So between the blood and air in the lungs would be outside, and blood and body cells would be for your tissues, which would be inside. Here's just an overview of that. You can look at it if you like pictures. So the two sides of the heart, you have the right side receives deoxygenated blood. That's the important part, is that the right side is always going to receive deoxygenated blood and then pump it to the lungs where it will become oxygenated, it'll offload its carbon dioxide, and then the left side receives oxygenated blood and pumps it to the body. That's always going to be true. Right is deoxygenated, left is oxygenated. The heart has four chambers. You have your atria, which are superior chambers that receive the blood, and ventricles that are inferior chambers that pump it away. So you can divide your heart into left and right sides and to superior, inferior sides. The right side is always going to be have deoxygenated blood. The left side is always going to have oxygenated blood. The superior chambers are always going to receive. The inferior chambers are always going to pump. There's that. So the great vessels are the ones directly attached to your heart. They transport blood to and from the heart's chambers. You have the pulmonary trunk, which is going to transport blood from the right ventricle. So remember, right ventricle, so it's still going to be both on the right side, so it's deoxygenated, and it's the ventricle, so it's low, so it's the inferior portion, so it's going to be pumping. So it's pumping deoxygenated blood. It splits from the pulmonary trunk, it splits into the pulmonary arteries. So arteries going away, remember it's from the right side of the heart, so it's deoxygenated. So here we have arteries that are deoxygenated. They'll go into the lungs. You also have the aorta, which transports blood from the left ventricle, so it's left side, so you know that it's oxygenated. And it's the ventricle, so you know that it's pumping it out of the heart. Your superior vena cava and inferior vena cava, these are your veins returning directly into the heart. They're draining deoxygenated blood into the right atrium. So it's deoxygenated blood, so you know it's going to the right side, and it's coming into the heart, so you know that it's going to be an atria, because it's going to be a superior chamber. The next are the pulmonary veins. They're draining deoxygenated blood into the left atrium. I'm sorry, oxygenated blood. I know that sounded weird. They're draining oxygenated blood into the left atrium. So you have from your pulmonary trunk coming right from your heart, you have your pulmonary arteries delivering deoxygenated blood to your lungs. That blood will come back via the pulmonary veins, where you're going to have oxygenated blood into the left atrium of the heart. Again, left side oxygenated, atria is receiving. There's the great vessels. The heart has two sets of valves. You have AV valves and semilunar valves. They're there to make sure your blood only flows one way in your heart. It can only flow one way. It needs to constantly flow that way, so the valves ensure that. The AV valves, the atrioventricular valves, sit between the atria and the ventricles of each side. If you only learn the abbreviation here, you may at some point forget that it stands for atrioventricular, and if you do, then you are going to be missing big hints for where these valves are. So remember that this is the atrioventricular valve. If you use AV, that's totally fine. I'm okay with that abbreviation, but just remember which ones they actually are. You have a right and a left AV valve. Your right AV valve is also your tricuspid valve. Your left AV valve has two different names, a bicuspid or a mitral valve. I will take any of them, that's totally fine. But again, I really like the right AV and left AV because it's very descriptive. And tricuspid and bicuspid, that refers to the papillary muscles on the valves. So you have to remember the structure for that, whereas for this, you just have to remember what AV stands for, and then you'll always know where they are. So this is what I prefer, but use any of them. The semilunar valves are the other ones. These sit at the boundary of ventricles and the arterial trunk. You have the pulmonary and an aortic semilunar valve. They're going to go exactly where you think. They're going to be gateways exactly where you think. The pulmonary semilunar valve is located between the right ventricle and the pulmonary trunk. So again, right ventricle, deoxygenated, pumping it out of the heart. It's going to go through that pulmonary semilunar valve into the pulmonary trunk, out the pulmonary arteries, to the lungs, back into the pulmonary veins. And then you have the aortic semilunar valve, which is between the left ventricle and the aorta. It will be pumping oxygenated blood out of that heart through the aortic semilunar valve into the aorta, which has several portions to it. Those are your valves. Okay, looking at pulmonary and systemic circulation. So we have two different circulatory routes. You have your pulmonary circulation, which is carrying deoxygenated blood from the right side of the heart to the lungs. And you have, then at the lungs, it picks up oxygen, it releases that carbon dioxide. And then you have your pulmonary veins bringing blood to the left side of the heart, oxygenated blood to the left side of the heart. By the way, you don't get rid of all of your carbon dioxide. You get rid of most of it, but not all of it. The systemic circulation, your oxygenated blood from the left side of the heart to systemic cells, meaning like the tissues, the body tissues, your arms, legs, organs, all those. The systemic cells, you're exchanging, let's see what that mark, yeah. Blood exchanges gases, nutrients, and wastes. You have your blood vessels return blood to the right side of the heart then, that's full of carbon dioxide and less oxygenated. But again, it does not offload all of its oxygen. It offloads about 25% of its oxygen. We'll talk about that. And then the basic pattern for circulation is going to be from the right side of the heart to the lungs to get oxygenated, returns on the left side, oxygenated. It goes out to its systemic tissues, it goes back to the right side of the heart, where it then goes back to the lungs to get for gas exchange. Here is this cardiovascular system route. You should be intimately familiar with this. You should be able to look at this and talk about any stages. You should be able to discuss, at the end of this, you'll be able to talk about the actual relative concentrations of oxygen and carbon dioxide, depending on where it is, whether it's in the lungs or the tissues. We'll get into it more. Okay, so congestive heart failure, it's impaired ability of the heart to pump blood. It can come from edema, which is a swelling, is this characteristic symptom of congestive heart failure. You have systemic edema, which can occur if the right ventricle is impaired. We have more blood remaining in systemic circulation and additional fluid entering your interstitial space, because as you have fluid remaining in your vessels, it will eventually start to pump out into the interstitial fluid, which can overwhelm your lymphatic system. Your pulmonary edema may occur if your left ventricle is impaired. So systemic is right ventricle, pulmonary is left ventricle. You have more blood than remaining in pulmonary circulation, swelling and fluid accumulation in the lungs, and it makes breathing difficult and impaired gas exchange. Okay, location and position of the heart. I'm not going to get too in-depth with this. This is lab stuff. You still need to know it, but you're going to go over a lot of the structural stuff in lab. So the heart sits posterior to the sternum, just left to the of the body to midline, so it's not right over your chest where you would maybe think, it's just a little bit to the side. It's between the lungs and what's called the mediastinum. So each lung is in its own sac, and then your heart has its own chamber as well, and it's called your mediastinum. It is slightly rotated. The right side is a little bit more forward than the left side, so it's just twisted a little bit towards the left. The base is the posterior superior surface, and the apex is the inferior conical end. So this will help you with lab a little bit. The base is not the bottom of your heart. The base is actually the back, the posterior superior surface. So it's actually on the back of the left ventricle. You have this kind of flattened portion of your heart, and that's the base. The apex is the pointy bit at the bottom. So yeah, you can see here, there's the apex there, and then the base would be posterior back here. And there is just a superior view of that. You can see each lung is in its own sac, and then you have your heart in its sac as well. Okay, the pericardium. This is the layers that enclose the heart collectively, so peri, around, cardium, heart. You have the fibrous pericardium, and then the parietal and visceral layers. The fibrous pericardium is the outermost covering of your heart, and it's exactly what you kind of think. It's just kind of, you'll see it when you do your APR lab stuff, but it's essentially just like whitish kind of connective tissue that covers the heart that you have to cut through to actually get to what you would think of when you see the heart, and usually what we picture, or what we depict. So it's dense, irregular connective tissue, attaches to the diaphragm and the base of the aorta, as well as the pulmonary trunk, and it anchors the heart and prevents its overfilling. So it provides a boundary so the heart doesn't overfill and stretch out. Okay, so then you have, that's the fibrous pericardium, the outermost covering. Then you have your parietal and visceral layers of the serous pericardium. So remember, the serous membrane is just an internal mucous membrane. So your mucous membranes in your nose and your genitals, your rectum, those are the mucous external membranes. Your serous membranes are internal. So you have the parietal layer, which is always going to be on the structures around it, and the visceral layer, which is always going to be directly attached to the organ. So the parietal layer attaches to this fibrous pericardium. It's kind of you have the fibrous pericardium on the very outside, then just inside to that you have the parietal layer, and then just deep to that you have this visceral layer of the serous pericardium. I'm not my finger over today. No, okay. You have the visceral layer just attached directly to the heart. The two layers are continuous and separated by the pericardial, you can kind of see it through me, pericardial cavity. So because you have two parts of this membrane, the parietal and the visceral layer, there is a small space between them, and that is this pericardial cavity. And here you go, you can see it. So this is the myocardium of the heart wall. So this is the actual heart itself. You have the endocardium, myocardium, and then this is the serous pericardium, which is also the epicardium. Okay, so the serous pericardium and the epicardium the visceral layer of the serous pericardium and the epicardium are exactly the same thing. Then you have this space right here, and then you have the parietal layer of the serous pericardium, followed by the fibrous pericardium. In that space, there is serous fluid, which acts as a lubricant, and the fibrous pericardium and parietal layer together form the pericardial sac, which holds the heart. Here you can see your pericardium. It's always hard in this. There we go. The pericardium wrapped around it. There's the fibrous portion, this space with serous fluid, and then you have that visceral layer attached to it. And then you can see just there is the myocardium, and then just lining that internal surface of the heart is the endocardium. Okay, pericarditis. So that is inflammation of the pericardium caused by viruses, bacteria, fungi. You have increased capillary permeability. Whenever you see inflammation, you should think capillary permeability, increased capillary permeability. Ultimately, you have fluid accumulation in the pericardial sac. That becomes eventually a real space. Once it fills with fluid, it starts to, before it's just kind of a potential space. It's there, but it's not really that large. This is definitely an exaggeration of how thick that actually space is. But once you have fluid in there, it starts to become a real space, and it can restrict the heart's movement and keep the chambers from filling, which results in cardiac tamponade, which is the heart unable to pump. You can have a friction rub, which is a crackling sound heard with the stethoscope as those pericardial layers rub together. That's just the external view of the heart. I think there's a couple of these slides, yeah. Okay, so now the layers of the heart wall. So the heart wall varies in thickness. With your ventricles, those are your pumping chambers, so those are going to be thicker. They're going to require more musculature. The left ventricle has a thicker wall than the right ventricle. Left must generate high blood pressure to force through systemic circulation. The right just pumps to nearby lungs. So your right side of your heart, you can see, is very, very thin right through here, whereas your left side has this thickened heart wall because it's pumping all the way out to our systemic body. The three layers of the heart wall: your epicardium, myocardium, and endocardium. So epi is the outside, the visceral layer of the serous pericardium, same thing. The outermost layer, simple squamous aerial connective tissue. The myocardium is the middle layer. It's the thickest, and it's that cardiac muscle tissue that contracts to pump your blood. And then your endocardium covers the internal surface of the heart and external surface of the valves. And it's continuous with the lining of blood vessels, of the vena cava and the aorta, as well as the arteries and veins coming to and from the heart. There's just another depiction of the heart wall. Again, you can see this thickened myocardial layer with that cardiac muscle tissue. There, again, is just another depiction. You can see this thickened side of the left ventricle that's pointing towards the apex. Okay, the separation of the chambers is done by two septum. Septum are those little, it's like the thing in your nose. If you see a close-up video of me, I have my septum pierced. That's what that is. The interatrial septum is between the atria. The interventricular septum separates the left ventricle from the right ventricle. Your right atrium, just a little bit more specific features of that. You have your pectinate muscles, which are the ridges on the anterior wall and within the auricle, which is a little flap that covers the outside of the heart. You have your fossa ovalis, which is an oval depression on the interatrial septum. If you remember from chapters, 7, 8, 9, skeletal stuff in 168, a fossa is a flattened portion of your bone. You have your temporal fossa, you have your iliac fossa on your hip, and then ovalis, meaning oval. So it's an oval depression, an oval flat spot on the interatrial septum on the right, on the right side. It occupies the location of what was called the foramen ovale. You'll see that when you do fetal structures. So foramen is a hole, so it's the oval hole, the oval window is what they call it. It eventually becomes the fossa ovalis. This, when we get into pulmonary circulation, we'll talk about it, but essentially because the fetus does not oxygenate its own blood, it just receives oxygenated blood from the mother. It does not have to shunt it out to the lungs, so there's this hole that allows blood to come into the right atria and then go straight over towards the left atria, down to the ventricle, and then out to fetal circulation. It's okay if you don't have to remember that right now. We will go more in depth into that. Your blood enters from the coronary sinus, carrying blood from heart wall. The coronary sinus carrying blood from the heart wall. So we'll talk about the vessels that actually surround your heart, your coronary vessels that actually bring fresh oxygen and take away waste to the heart itself. Because not only does the heart have to pump blood out to the body, it also has to supply blood for itself. The veins that supply, that drain waste from the heart all empty into what's called this coronary sinus. You also have your superior vena cava and inferior vena cava entering, all of this going into the right atria. It exits to the right ventricle through the right AV valve. And then we're in the right ventricle. You have your papillary muscles, which are cone-shaped projections extending from the internal ventricle wall. The right side typically has 3 of them, again, that tricuspid valve. And so those are the actual muscles projecting from the heart wall. Then you have your tendinous cords, your chordae tendineae, which are thin strands of collagen fibers attaching to the AV valve. So the tendinous cord, the papillary muscles come from the heart wall. They, in turn, are attached to the tendinous cords, which then attach to the AV valve to help anchor it. That's what these do. The superior exit to the right ventricle is to the pulmonary trunk through the pulmonary semilunar valve. So you enter the right ventricle through the right AV valve, the tricuspid valve, and then you exit it through the pulmonary semilunar valve. Then when you come back, so that's all the lung stuff, and then you come back into the left atria, which has entrances from pulmonary veins. It exits to the left ventricle through the left AV valve. Once you get through the valve, you're in the left ventricle. You have 2 papillary muscles that anchor the chordae tendineae on the left side. So that's why it's also called your bicuspid valve. And the superior exit to the aorta through the aortic semilunar valve. So the semilunar valves are going to be leaving the heart. The AV valves regulate movement through it. There's some internal anatomy if you want to look at it. That little hole is the coronary sinus. This larger one is the fossa ovalis. You will need to know those for lab. All right, the heart valves ensure one-way blood flow. 2 types: atrioventricular, semilunar. The AV valves prevent backflow to the atria from the ventricles. Your right AV is between the right atria and ventricle, also your tricuspid. The left AV is the mitral, between the left atria and ventricle, also called your bicuspid. The AV valves close when the ventricles contract so that to force the blood superiorly, and the papillary muscles and tendinous cords prevent inverting into the atria. So what's happening is that when the ventricles contract, as the ventricles contract together, they're going to be pushing blood up and out. The right side is going to be going out through the pulmonary semilunar valve. The left side is going out through the aortic semilunar valve. But to make sure that the blood only goes out through those valves, the AV valve separating the ventricles from the atria, they have to lock down. So the papillary muscles that extend from the ventricle heart wall that go to the tendinous cords, those attach to the valves and they help to anchor it down. So as blood is getting pushed out, it's hitting the AV valves, but because of how they're shaped, they're kind of shaped down like this. So when blood hits them, they actually can close up. The papillary muscles help to anchor the tendinous cords to keep it closed. And so then blood only can go out through the semilunar valves to the lungs or to the body. The semilunar valves permit backflow into the ventricles from the pulmonary trunk, from the aorta back into the ventricles. They open when the ventricles contract and blood is forced into the arteries. They close when the ventricles relax. Your arterial pressure becomes greater than ventricular pressure. As blood starts to slide backward, it catches the cusps and closes valves. So once we start getting into heart stuff, we do start talking about pressures, and that can get a little confusing for people. So arterial pressure becomes greater than ventricular pressure. The pressure that it's talking about is the pressure from the force of the ventricles contracting and pushing blood out, as well as the force of blood that's in the aorta pushing back against that AV valve. Because there's already blood in the aorta. As soon as your heart contracts and pushes blood out there, it's just pumping more blood in there. There's already blood kind of sitting back there. Because what happens is once the ventricles contract and they've emptied out of their blood, they're going to start to relax. And when they start to relax, all of that blood that's sitting in your aorta, it'll start to kind of slide back down and it'll push against these pulmonary semilunar valves. But they catch because the way that the aortic semilunar valves, but because of the way that they're shaped, kind of cupped down like this, as blood flows back down, they kind of get stuck shut. Yeah. That's how both of the valves function. There's your valve locations. Okay, heart valves, the mechanism of action. This APR, every time you see this, that is on Connect. So APR is Anatomy Physiology Revealed. There are videos on that once you launch that application. And there's one on the cardiac cycle. It gives you a 3D view through it. Highly recommend you watching that. I'll try to, it's easier for me to share the screen on the Teams application. So tomorrow, on Tuesday, when I do my lecture kind of quote unquote portion, I will try to go over these videos and try to remind all of those videos. Okay, so for the atrioventricular valve, it's opening. So you can see you have 1, 2, 3 cusps here. So you have blood flow coming in. It's going to be opening. That flow of blood is going to help to open up the valves. And then in this case, once the ventricle gets filled, the pressure in the ventricle will be great enough to close that valve. So in this case, for here, when you're flowing down from atria into ventricle, the pressure in the atria here is greater than the pressure in the ventricle. Once the ventricle fills, the atria is emptied. Now the pressure in the ventricle is greater than the pressure in the atria, and so the valves close. For the semilunar valves, you have the pressure in your ventricle is overcoming the pressure in your artery to open them. And then once those ventricles are emptied, the pressure in the artery is greater than the pressure in the ventricle, and so it closes the valve. Okay, teenage athletes in sudden cardiac death, just to really take a 90-degree turn here. Sudden death caused by undetected cardiovascular disease, most due to congenital heart defects and coronary artery anomalies. Again, the coronary arteries are the arteries that surround and deliver blood to the heart. It's a result of cardiomegaly, which is an increased thickness of the heart, the symptoms of swelling, dizziness, arrhythmia, and shortness of breath. It can be revealed by a standard X-ray and confirmed with an echocardiogram. Heart sounds and heart murmurs. So there are 4 normal heart sounds, the 2 familiar ones, the lub-dub, lub-dub. S1 and S2 is what we call those. S1 is the closing of the AV valves, and S2 is the closing of the semilunar valves. So lub-dub, lub-dub, AV, semilunar, AV, semilunar, or I guess, yeah, AV, semilunar, AV, semilunar. And so it's the sound of the valves closing. Your 2 minor sounds are S3 and S4, and these are clinically useful. They are important. Each valve is heard best at specific anatomic locations. That's why you kind of, they do a couple of different places. And if you have a heart murmur, that is an abnormal heart sound, which is a result of turbulence of blood passing through the heart. Some medically important, some not. And there are two types of valvular insufficiency and valvular stenosis. So typically, even though it is a fairly, there's a lot of pressure being generated in the heart, it should be a fairly smooth process. But sometimes when you have valves that are either don't close properly, which is a valvular insufficiency, or valvular stenosis, meaning that they don't open properly, that can then cause the blood flow to be turbulent, because instead of having these nice smooth exits and entrances, it's kind of pushing up against things, or it's being able to go through retrograde and actually be able to go maybe from the ventricle back up into the atria if the AV valve isn't closing properly. The valvular insufficiency, I don't know why I talk ahead. The valvular insufficiency, cardiac valves leaking because cuffs not closing tightly. Again, that's the AV valves or the simulator valves not closing properly. Blood regurgitates back through the valve, and you may get heart enlargement because of that, because the heart's having to work harder to try and pump blood out of it, because the heart can detect the stretch. It doesn't detect, like the liquid, but it detects the stretch of having blood remaining in it. Your valvular stenosis is when the valve cup's scarred and cannot open completely. It presents resistance to blood flow, reduces the chamber output, so how much blood your heart is actually putting, and again, the chamber may enlarge and dilate to try and push more blood out through these hardened valves. Okay, microscopic structure of cardiac muscle. So the myocardium, that's your muscle layer of your heart, is composed of cardiac muscle tissue. Your cardiac muscle cells are short and branched. Always just make sure that I'm still recording. They house one or two central nuclei, and the outer covering of those muscle cells is called the endomysium. Okay, the endomysium is the outer covering of those muscle cells. Your sarcolemma is the plasma membrane, if you remember, for your muscle cells. It invaginates to form T-tubules extending into the sarcoplasmic reticulum. So hopefully you remember all the stuff from muscle contractions. The sarcoplasmic reticulum surround the bundles of myofilaments, and they have the calcium. The myofilaments are arranged in sarcomeres, just like in skeletal muscle. The organization gives rise to striated appearance under microscope, and the optimal length is what's called the maximum overlap of filaments is seen when the heart is stretched by the blood filling the chamber, which allows the filaments to contract with greater force with more blood. Essentially, so if you remember, hopefully you have refreshed yourself on your muscle physiology. But you have your sarcolemma, which is your plasma membrane, and then along the plasma membrane, it goes down into the muscle cell. It actually invaginates and penetrates into the muscle cell. Those are the T-tubules. And they extend into the sarcoplasmic reticulum, which surrounds the myofilaments, and it's going to have those terminal cisternae, which house calcium that will then be released into the myofilaments for muscle contraction to happen. This optimal length, remember your myofilaments, your actin and myosin. This optimal length, what it's saying is that your heart, when it's empty, when it doesn't have any blood in it, in the ventricles rather, or in whichever chamber, because it's a boom, 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 boom kind of thing. So when the chambers don't have any blood in them, when they're not being stretched, they're at the relaxed point, that's not going to be their optimal point of contracting. The muscles in the heart aren't going to be able to contract until they've been stretched a little bit, until you've kind of they started to actually fill with blood. That's when the filaments will kind of reach their optimal overlength to be able to contract more fully. Up until that point, they're the heart muscle cells. So with normal skeletal tissue, at rest, our muscle tissues or sarcomeres are probably about that overlapped. So then we can contract immediately. So we can go from rest to immediate contraction. With our heart, it rests more overlapped. So the myofilaments are going to rest more there. And so then when it gets filled with blood and it stretches, that's when it's then going to be able to contract more forcefully. The intercellular structures, your sarcolemma is folded at connections in between the cells. This increases structural stability of the myocardium and facilitates communication between the cells. Important part, though, is the intercalated discs. The intercalated discs are two structures. It is your desmosomes and your gap junctions. So these are individual things. Collectively, they make up your intercalated discs. The desmosomes mechanically join cells with protein filaments, so this is a physical hole between one cardiac muscle cell and the next one. And the gap junctions electrically join cells, which is allowing ion flow to make sure that each heart chamber is a functional unit, is a functional syncytium. We'll talk about this. This is one of my favorite words, syncytium. It's just fun to say. So these desmosomes are going to mechanically join them. The gap junctions allow that ion flow to move through. So that way your atria contract as one, and then your ventricles are going to contract as one. What syncytium means is that you still have individual heart cells. You can still see the differentiation between the individual heart muscle cells, but they're going to act as one. So as soon as one heart cell gets stimulated, all of the ones that it's connected to via these gap junctions is going to function together. It's going to depolarize as well. Here is just a cross section of that. You can see the intercalated discs, and here your desmosomes, and then your gap junctions. So again, these are physically linking them, and these are creating ion pores. There is just the structural view of your cardiac muscle cell. You can see your sarcomere here. Remember, the sarcomere is the unit of contraction. It'll go from Z disc to Z disc, and that shortens, and as that shortens the muscle cell. Okay, metabolism of cardiac muscle. It has a high demand for energy, as you may imagine. There's extensive blood supply through your heart with numerous mitochondria. The heart has what's called myoglobin and creatine kinase. The creatine kinase transfers phosphate from creatine to make ATP, so that's kind of like a backup emergency supply of energy. And myoglobin, that is like hemoglobin, except it's in your heart muscle. It can only carry one oxygen though, whereas hemoglobin can carry 4 oxygens, myoglobin can only carry 1 oxygen, but there's a ton of it, so it makes up for that. The heart is able to use different fuel types, different fuel molecules, so fatty acids, glucose, lactic acid, amino acids, and ketone bodies. It will convert all of those to the glucose if it needs to. Mostly it relies on aerobic metabolism. You breathe, your heart relies on that oxygen to create energy. This makes it susceptible to failure when ischemic, when the oxygen is low. An interference with blood flow to normal heart muscle can cause pretty rapid cell death. So even though it can use lots of these energy sources, it takes time. It takes time to convert fatty acids or lactic acid or amino acids into glucose. And so in that time, you're going to be having ischemia. You're going to be having low energy failure or oxygen failure and metabolic failure in your heart cells, which can lead to death. All right, finally, coronary circulation. This is delivering blood to the heart's wall. The coronary arteries transport oxygenated blood to the heart wall. Coronary veins transport deoxygenated blood away from the heart wall towards the right atrium. Again, I'll let you remember what structure from the coronary veins into the right atrium. I'll let you remember what structure it's going to pass through to empty into the right atrium. There's just a depiction of that. You can see they kind of wrap around and then down the heart. All right, your coronary arteries are functional end arteries. So although their structural anastomoses exist, I'll talk about that in a second, blockage of a coronary artery leads to insufficient flow of the heart wall. So an anastomosis, what is this even saying, right? So an anastomosis are connections between vessels allowing blood to arrive by more than one route. Essentially what's that saying? It's this is a fail-safe, basically. So if one of the arteries is blocked, there's an anastomosis. There's another artery that could just kind of divert the blood flow to that will deliver it to the same structures. However, because the coronary arteries are so small, that usually when one is blocked, the anastomosis, it really doesn't matter. It's not going to be enough to allow blood to flow through. The coronary anastomoses are too small to function as effective alternatives, as effective routes around for blood. Your coronary flow is intermittent with the heart contractions. The vessels patent, which means open. So the vessels open when the heart is relaxed, and the vessels are compressed, interrupting the flow when the heart contracts. So when the heart starts to relax, the vessels can kind of open back up, and they'll allow blood to flow. And then when the vessels are, when the heart starts to contract, the vessels get contracted, and their blood gets pushed out. So it's pulsatile, is how we would say that. Blood flow is intermittent, it's pulsatile. Several cardiac veins drain the heart muscle. The great cardiac vein is in the anterior interventricular sulcus. Don't let names scare you. A sulcus is a kind of shallow groove. So the anterior on the front, interventricular, enter between the ventricles and the groove. So it's the groove in between the ventricles on the front. The middle cardiac vein sits to the posterior interventricular sulcus. It's on the back. The small cardiac vein sits next to the right marginal artery and the coronary sinus. That's in the posterior aspect of the coronary sulcus. If you did not answer my question from the other side, this is where the cardiac veins drain into. And there you go. So note here, the coronary sinus is a vessel as well as the hole that's in your heart that blood is going to drain into the right atria. So a sinus is both kind of is the passageway and the space itself. So coronary sinus refers to this large vessel as well as the little hole right there that empties directly into the right atria. Okay, atherosclerosis. Plaque is narrowing your coronary arteries. You can have a coronary spasm, which is a sudden narrowing of the vessels. A spasm is what you think about, like a little jump. Both of the above can lead to angina or myocardial infarction, angina being general chest pain. There you go, angina pectoris, usually on the left side of the chest, left arm or jaw, and usually referred pain when performing a strenuous activity. So if you're performing a strenuous activity, your heart may have to work more, and that will refer pain down to the left side of your body. A myocardial infarction is a heart attack. You then have sudden and complete occlusion of coronary arteries, so occlusion means blocking. The myocardium is deprived of oxygen, possibly tissue death if it's long enough. And you get excruciating chest pain radiating down your left arm, weakness, shortness of breath, nausea, anxiety, and sweating. This is, I would say, again, I'm not a clinical physician, but fairly out of date, especially when you update symptoms for women. They tend to present heart attacks in very different ways. So definitely, that's for men and for very general, I think. Okay, getting into the heart's conduction system. So as we'll learn in the heart through these next slides, there are electrical events and mechanical events in your heart. The electrical events are going to be this conduction system. The mechanical events are actually going to be the cardiac cycle, where we're actually pumping blood through the heart. But the conduction system is what regulates the mechanical events, the cardiac cycle. So conduction system initiates and conducts electrical events to ensure proper timing of contractions. You have special cardiac muscle cells that have action potentials but do not contract, and its activity is influenced by autonomic nervous system, your ANS. These are the components of your conduction system. Your sinoatrial node, your SA node, initiates your heartbeat. It's called your pacemaker. It's known as your pacemaker. You can have an artificial pacemaker put in. It does this. It mimics the activity of the SA node. The AV node is located in the floor of the right atrium, near the right AV valve. The atrioventricular bundle, the bundle of His, extends from the AV node through the interventricular septum and divides into the left and right bundles. Your Purkinje fibers extend from the left and right bundles at the heart's apex and course through the walls of the ventricle. So starting up in the superior portion, actually, here we go. So again, APR conducting system of the heart, there's a video that walks you through this, a very good video that walks you through this. Here is your heart, superior portion of the right atria. You have your SA node. The signal goes from there. It spreads actually from here through the right atria and to the left atria. So the atria are syncytium with each other. They are in syncytium with each other. So they contract together, and then your ventricles contract together. So from the once the SA node depolarizes and fires a signal, it's going to spread from here out through the right atria, across to the left atria, and then it's going to go into the AV node. At the AV node, it will then be transferred down through this bundle of His. It'll split into the left and right bundle branches. And then as it goes along the outer wall, it'll be the Purkinje fibers. And once it starts to go down through here, this is when, this is going to be the signal, this is going to carry the signal for the ventricles to contract. So the heart and respiratory rate are influenced by the medulla oblongata, just for remembering. Here is your pons, your medulla, and your spinal cord, part of your brainstem. The cardiac center of the medulla oblongata, it has the cardioacceleratory and cardioinhibitory centers. These are going to receive signals from baroreceptors, which are stretch receptors, and chemoreceptors, monitoring carbon dioxide mostly, in your cardiovascular system. These are going to send sympathetic and parasympathetic signals, modifying but not initiating cardiac activity. So the cardiac center, it's acceleratory, it's inhibitory. It is not actually giving the signal, okay, contract now, contract now, contract now. That's all happening at the SA node. What the cardiac center can do is say, contract faster, contract slower, contract slower. Or, I guess I should say, slower and stronger. So with parasympathetic innervation, that's going to decrease your heart rate. It's coming from the cardioinhibitory center through your vagus nerve, your cranial nerve 10. Your right vagus innervates the SA node. Your left vagus innervates the AV node. I'm not going to ask you that specifically, right and left vagus. Your sympathetic innervation increases the heart rate and force of contraction. So this starts at the cardioacceleratory center. It's going to increase the heart rate. Cardioacceleratory makes sense. It's relayed via neurons from the T1 to T5 segments of the spinal cord, so thoracic 1 to thoracic 5. Again, think about where your heart is and where those nerves innervating it are. Sympathetic, it's not the vagus nerve, it's parasympathetic. Extends to the SA node, the AV node, the myocardium, and the coronary arteries, increasing coronary vessel dilation. If you increase coronary vessel dilation, you're going to increase coronary blood flow, which will allow for more oxygen delivery, more carbon dioxide removal, to allow for a stronger heart. Here is just a graphic representation of all of that. Again, your baroreceptors are detecting the stretch of the heart, so that's going to help you with your force. The chemoreceptors are detecting mostly your carbon dioxide concentrations to help you with the rate of how fast you need to be pumping. Heart contraction involves two events. Your conduction system initiates and propagates the action potential, and the cardiac muscle cells fire action potentials and contract. The process happens first in the atria and then in the ventricles. Okay, so the initiation, the SA node initiates the action potential from one of these nodal cells, and again, it starts there and it spreads down and across the left atria. The action potential is spread through and propagated through. And then the cardiac muscle cells, the action potential is propagated along the sarcolemma of the cardiac muscle cells. And the muscle contraction allows the thin filaments to slide past the thick filaments, and the sarcomeres shorten within the cardiac muscle cells. That's kind of your basic overview of what's happening. All right, these nodal cells in the SA node initiating your heartbeat, they're going to spontaneously depolarize and generate action potential. Spontaneously, meaning that they just do it on their own. They have these leak channels that are going to make sure that they just keep depolarizing. If you remember depolarizing, you have polarize, which means that it has a charge, a negative charge to it, and we're depolarizing, so we're removing the negative charge to generate that action potential. For heart cells, the resting membrane potential is about negative 60 millivolts, but the cells do not have a stable resting membrane potential. They have many common membrane, that's probably like another line, not a stable resting membrane potential. They have a lot of membrane proteins, sodium potassium pumps, calcium pumps, and leak channels that help to maintain that resting membrane potential, as well as slow sodium, fast calcium, and voltage-gated potassium channels. Slow and fast means exactly what you think. Okay, so in your interstitial fluid, you have your slow voltage-gated calcium channels, fast, slow voltage-gated sodium channels, fast voltage-gated calcium channels, and voltage-gated potassium channels. The SA nodes exhibit autorhythmicity, meaning spontaneous firing. Again, the medulla oblongata does not initiate the pulse, the depolarization. The SA node does it itself. To reach threshold, you have these slow voltage-gated sodium channels open and sodium flowing into your nodal cell. Remember, it's negative. We're trying to depolarize, trying to get it more positive. So these sodium channels is flowing in, and then resting membrane potential changes from negative 60 to negative 40 millivolts. This is your threshold value. So just as after, let's say we're right at the beginning of one of these cycles, the cell is just at rest, it's at negative 60 millivolts, and as soon as it gets to negative 60 millivolts, the slow voltage-gated sodium channels are open, and they're going to allow sodium to flow into the nodal cell, and it's going to go from negative 60 to negative 40 because sodium is positive. Once it gets to negative 40, that's its threshold value. You're going to then go into depolarization. You have your fast voltage-gated calcium channels opening. So they're voltage-gated, meaning they're going to open at a certain voltage, negative 40 millivolts. And they're fast. So they're going to open a lot of calcium. A lot of those positive calcium channels are going to open. Calcium is going to flow in. The membrane potential changes from negative 40 to just above 0 millivolts. And then you have repolarization. Your calcium channels close. So again, they're voltage gated. So once it reaches just above 0, your calcium channels are going to close. The voltage gated potassium channels open, so potassium can flow out of the nodal cell. Membrane potential goes back to negative 60 millivolts, and that's when again your voltage gated sodium channels open back up, and the process begins again. At rest, one SA node action potential starts about 0.8 seconds after the last. So less than 1 second later, you're having this depolarization process happening again and again. It's depolarizing and repolarizing in that time. Translates to about 75 heartbeats per minute. Inherently, the SA node would fire faster at 100 per minute. So what's happening is what's called vagal tone. So in the body, your vagal tone is your parasympathetic activity relayed by the vagus nerve, keeps the resting heart rate lower. So remember, the parasympathetic activity is part of your cardioinhibitory center, so it's going to be translating its signal through the vagus nerve. Your vagal tone is also what keeps your rectum just kind of closed and your external sphincters just inherently closed, and a slight tone to your abdominal organs, so that way you don't just use the restroom on yourself. So normally, just on its own, the SA node would fire at 100 beats per minute, but because we have this vagal tone, this constant parasympathetic activity coming, it goes down to about 75. The pacemaker potential of nodal cells is the ability to reach threshold without stimulation. So nothing is stimulating them. These nodal cells are pacemaker cells. So here, if you like a graphic depiction of that, you have your time versus voltage. Starting down here at 1, you're at your resting membrane potential at negative 60. Sodium starts to flow in. It reaches threshold. You have these fast voltage gated calcium channels that are going to open and depolarize the cell to just above 0. At 0, the calcium channels will close. The potassium channels will open. You have this potassium outflow to repolarize the cell. There. When it gets to the negative 60, the sodium channels will open again, and you go through all over again. This is your pacemaker potential. So this is the sodium channels. The sodium channels just allowing the cell to spontaneously depolarize. This is that threshold voltage that it has to reach. Once it reaches threshold, it's going to have an action potential. This is just, again, a different depiction of that. Okay, after starting at the SA node, the action potential spreads. So that was all just getting the action potential to initiate at the SA node. Once you have that, the action potential distributes through the atria, reaching the AV node. That excitation travels via gap junctions, and the two atria contract together. So again, this depolarization is going to travel through those ion channels, so that the two upper chambers can contract at the same time, pushing their blood down into the ventricles. The action potential is delayed at the AV node. Your AV nodal cells are slow because of the small diameter and fewer gap junctions. And this is good. The delay allows the ventricles to fill before they contract. So it allows this delay at the AV node allows the atria to fully contract and empty into the ventricles. Insulation of fibrous skeleton means the AV node is bottlenecked, so it's the only path. So it kind of just forces all the electrical signal. It can't just go through from the atria through the ventricles. It has to travel through that AV node, through that special pathway. The action potential travels through the AV bundle to the Purkinje fibers. So you have AV node to your AV bundle to your left and right bundle branches, and then your bundle branches to your Purkinje fibers, which spread along the heart wall, the external heart wall. The action potential then spreads through the ventricles. Gap junctions allow the impulse to spread through the cardiac muscle fibers, and the cells of the two ventricles contract nearly simultaneously. So just as the atria are going to contract together, the ventricles are going to contract together. Here we go. You can just see that in action. The action potential generated at this SA node there. It's spread via gap junctions between the cardiac muscle cells to the AV node. It's delayed at the AV node before it passes to the AV bundle in this interventricular septum right there. The AV bundle conducts it down the left and right bundle branches, the Purkinje fibers, and then it spreads to the ventricles, and so the ventricles can contract altogether. Right. Purkinje fibers are larger in diameter than other cardiac fibers. This allows the action potential to be extremely rapid. It goes very quickly, ensuring the ventricles contract at the same time. Your papillary muscles are stimulated to contract immediately, anchoring the chordae tendineae of your AV cusps, and pulling on the cusp just prior to an increase in pressure of the ventricles. So before the ventricles contract and can pump the blood, the papillary muscles are already kind of holding down those tendinous cords to brace it, so that way they don't get ripped off when the ventricles contract. Stimulation begins at the heart apex and ensures blood efficiently ejected toward arterial trunks. So even though they are contracting together, and I keep doing it like this, it really is going from the bottom kind of around and making sure that the ejection goes up and not just either down or sideways or whichever. It's actually going, the ejection is superior. Same thing with the atria. They contract almost instantaneously. Almost all cells contract instantaneously, except there is just enough delay to make sure that the atria contract inferiorly and the ventricles contract superiorly. An ectopic pacemaker is a pacemaker other than the SA node, so ectopic. Conduction system cells other than your SA node also have the ability to spontaneously depolarize. So other cells in this conduction system can act as the pacemaker. They depolarize at slower rates than your SA node, though. The AV node is a default pacemaker if the SA node is impaired with an inherent rhythm of 40 to 50 beats. It's fast enough to sustain life, so it'll work, but you'll be weaker. In cardiac muscle, fast enough to sustain life, that's not really encouraging. In your cardiac muscle, with a rate of 20 to 40 beats per minute, that is usually too slow to sustain life. So it's not great. That's it. Okay, electrical and mechanical events of the cardiac muscle cells. So remember, to have a mechanical event, you have to have an electrical event. Sorry, this might destroy everything. Okay, still standing. All right, your depolarization, your impulse from the conduction system. Your impulse from the conduction system or the gap junctions opens fast-gated sodium channels that sodium enters, changes the cell membrane potential from -90 to +30. So we are no longer talking about the nodal cells. We're no longer talking about the conduction system of the heart itself. We're talking about the cardiac muscle cells. I know it's hard to separate those two things, but you have your electrical system of your heart, and you have your electrical mechanical system of the muscle cells. So once the SA node, once that the conduction system of the heart starts to depolarize, it'll transfer that depolarization, that signal, to the cardiac muscle cells. So you have still the same opening of, this time, fast voltage-gated sodium channels, not slow but fast. Your sodium enters the cell, changing the potential from -90 to +30. So your cardiac muscle cells, they rest at -90. The voltage-gated sodium channels start to inactivate once they get to the +30. You then hit a plateau. The depolarization opens voltage-gated sodium and slow voltage-gated calcium channels. The sodium leaves cardiac muscle cell as calcium enters and stimulates sarcoplasmic reticulum to release more calcium, and your membrane remains depolarized. So you have all of this sodium coming in and immediately just jumping and depolarizing your cell membrane. Then once it gets to +30, the voltage-gated turn off, and you have these other two voltage-gated channels that are going to open simultaneously. Potassium is going to be leaking out, trying to make the cell more negative, the heart cell more negative, whereas calcium is going to be coming back in, trying to keep it depolarized and causing this plateau. After that, you then have repolarization. Your voltage-gated sodium channels close while potassium channels remain open, and the membrane potential goes back to -90. Here is a depiction of this. So starting down here at -90, you have that stimulus coming from the conduction system of the heart, the SA node, the AV node, the bundle, the Purkinje fibers, whatever it may be, or just gap junctions, who knows. So it's coming from this resting membrane potential. The fast voltage-gated sodium channels open and depolarize the cell. Once it gets to +30, these voltage-gated channels close off, sodium stops entering, and you have your potassium-calcium channels opening. You're going to have potassium out, sodium in, or calcium in. That's going to delay the repolarization into this plateau period until eventually you have the calcium channels close and the potassium channels stay open. Here is just another prediction of that, or depiction of that. Okay, now the mechanical events of the heart cells, the cross-bridge cycling of the heart cells. So you have the electrical events of the conduction system in the heart, you have the electrical events of the cardiac muscle cells, and now the mechanical events, the actual contraction of the cardiac muscle cells. The calcium enters the sarcoplasmic reticulum from the interstitial fluid and sarcoplasmic reticulum, leading to contraction. So the sarcoplasm, if you remember, is just the cytosol of the cytoplasm of the heart muscle cell. So calcium coming in is going to be floating around inside of the fluid into the sarcoplasm. And as in skeletal muscle, it binds to troponin and initiates cross-bridge cycling. You have cross-bridge formation, which is where the myosin attaches to the actin. You have power stroke, where myosin pulls the actin, and the release of myosin heads and the reset of the myosin head. Remember, it takes ATP to reset the cells. It does not take ATP to contract, it takes ATP to relax. The calcium levels decrease, leading to relaxation. Calcium channels close and pumps move it out of the sarcoplasm, out of the cell cytosol, into the sarcoplasmic reticulum and out of the cell. Okay. Cardiac muscle cannot exhibit tetany. Tetany is prolonged contraction. Unlike skeletal muscle, cardiac cells have a long refractory period, that plateau, where the calcium and the potassium are going in and out. That causes a refractory period. The cell cannot fire a new impulse during the refractory period. And cardiac muscle cells plateau phase leads to that. It's about 250 milliseconds. The heart cell contracts and relaxes before it can be stimulated again and makes sustained tetanic contraction impossible. So you can clench your jaw, you can kind of flex your elbows and clench your hands, and you can maintain that for a long time if you're not holding anything. You can kind of keep your muscle contracted. I can stand up for hours and hours. So we can have these prolonged contractions. In humans, we don't actually have true tetany. We can't truly lock up unless you have some kind of virus or bacterial infection. But it is pretty much the same. We can kind of lock our muscles. The heart can't do that. Because of the plateau period, because of this refractory period, you have this delay before the muscle can be contracted again. So this is just for skeletal muscle cells. You see that you can have a electrical event and then a corresponding contraction, or you can have this prolonged frequency of stimulation. You can have a lot of nervous stimulation leading to this prolonged, eventually constant contraction until you have muscle fatigue. For heart cells, because they have this prolonged plateau phase, their contraction is a little bit longer, and you can only stimulate it so many times. It just does not have the ability to depolarize so rapidly to allow for this kind of rapid stimulation. That essentially just makes sure that the heart empties and fills properly. Oh wait, electrocardiogram, ECG, EKG, same thing, one's Greek, one's Latin. The skin electrodes detect electrical signals of the cardiac muscle cells, a very common diagnostic tool. You have different waves and segments. The P wave reflects the electrical changes of atrial depolarization originating in the SA node. The QRS complex is the electrical changes associated with ventricular depolarization. Also simultaneously, the atria is repolarizing during this. And then your T wave is associated with ventricular repolarization. So atrial depolarization, ventricular depolarization, and atrial repolarization, and then ventricular repolarization. So you can essentially think about this as atria contracting, and then ventricles contracting, atria relaxing, and then ventricles relaxing. The two segments between the waves correspond to plateau phases. Your PQ segment is the atrial's plateau, and the SC segment is the ventricular plateau. And that's not so. The plateau isn't when they're resting. The plateau is when they're contracting to make sure they empty fully. All right, so here is just a picture of that. You have the P wave, which is again that atrial depolarization, atria contracting, ventricular depolarization, and in here, in this QRS complex, this is when you also have your atria repolarizing so that they can then contract again at the end of the cycle. But during this, again, it's such a big peak because that's your ventricular depolarization. So the ventricles are stronger. They have a big kind of lot more pumping to do. This ST segment, that's the ventricles contracting, and the T segment, that's the ventricles repolarizing. The PR interval is the time from the beginning of the P wave to the beginning of the QRS deflection, from atrial depolarization to ventricular depolarization, and it's the time to transmit action potential through the entire conduction system. Your QT interval is the time, transmit action potential through the entire conduction system. That means AV node to Purkinje fibers, atrial depolarization to ventricular depolarization. The QT interval is from the QRS to the end of the T wave, and it reflects the time of the ventricular action potential. So the QRS to the end of the T wave is a full cycle of your ventricles, full depolarizing repolarization cycle. The length depends upon your heart rate, and changes may result in tachyarrhythmia, a rapid irregular heart rate. Okay, so this is comparing the electrical changes of the atria with the electrical changes of the ventricles. Again, just kind of noting how much smaller that atrial signal is. I don't know why I feel like I need to trace it with my finger, but how much smaller that atrial signal is compared to the ventricles, because the ventricles have a much larger job to do. All right, this is always overwhelming. Don't worry. I usually have to look at it a couple of times, like for a second or two, before I can really start making sense of it. But this is one of those charts. You have a similar one in your book it has like a second portion to it with the cardiac cycle in it. But yeah, if you can look at this and you can understand what's happening, you can talk your way through this, you pretty much understand this whole chapter. So that's what's cool about this image. As overwhelming as it can kind of be, if you can talk your way through this chapter, or through this image, you can pretty much do all the stuff that we have in this chapter. So a couple things to look at. Down here, you're measuring a lot of different things. So down here, you have the volume changes in your atria and ventricle. And then up here, you have your pressure changes in your atria and your ventricle. It also talks about the different phases of the cardiac cycle. Okay, so we'll kind of go slow with this. This is your electrocardiogram, your EKG. So you can see your P, Q, R, S, T waves. And they have them labeled on here, P, Q, R, S, T. If we follow this, we follow P, that is the atrial depolarization. So this is when our atria are going to start to depolarize, and then right here is where our atria contract. Now if we go up, right here, we're looking at ventricular volume. So when our atria are contracting, we see this simultaneous spike in ventricular volume. It's because the atria are contracting and pumping blood into the ventricles. If you keep on going up, you see this corresponding spike in atrial pressure and ventricular pressure. The atria are contracting, their pressure increases. The ventricles are filling with blood, their pressure is starting to increase. As we keep kind of going, I'm sorry, that's that pressure. Right here, the aortic pressure is dropping because if the ventricles are filling with blood, that means that they're relaxed. And so this means that they're not pumping blood into the atria. This means that into the aorta. So this means that the blood in the aorta is starting to relax. So you have an increase in ventricles, an increase in atria, but a decrease from the aorta. Now if we go, we have QRS. So this is when the ventricles are starting to depolarize. So we can see that during that ventricular depolarization, there's no change in volume. But then right up here, so this is where that R, if we follow that R up, you see this immediate big spike in ventricular pressure, okay? This spike in ventricular pressure corresponds with this dip in ventricular volume. Here is our contraction phase for the ventricles. So here's our electrical signal saying, all right, now it's time to fire. You have this immediate increase in pressure. And then look at this little spike right here. So there's this line, the solid line, and then this dotted line. We call that the isovolumic contraction. So this little space right here is actually significant. It's saying, okay, this solid line is when the signal is delivered to contract. And this is the time that it takes for that signal basically to reach to the muscle cells, and this dotted line is then when the contraction actually starts. Or I'm sorry. this is when the solid line is when the signal goes to the cardiac muscle cells in the ventricles. The ventricles start to contract, but you have this moment of when the ventricles are contracting, they're fighting against this aortic pressure. So in this little space right here, you have the ventricles full of blood starting to contract, fighting against the pressure from the aorta. And then right at this moment right here, that's when they overcome aortic pressure, and you see, again, no change in volume from this space, but then as soon as they hit the dotted line, you see this drop in ventricular volume. That's the ventricles emptying. Once the ventricles empty, the ventricles then repolarize. At the end of repolarization, you start to then see this, increase in ventricular volume because we're coming back here. We're going from T into the start of the cycle, the atria contracting and filling the ventricles. So if you can, it's a lot. So if you can read through that and you can work through your way through that a lot, this is a great study tool for everything we're doing. All right, cardiac arrhythmias, you can have heart blocks, impaired conduction. A first degree is a PR prolongation, slow conduction between the atria and the ventricles. Second degree AV block is a failure of some atrial action potentials to reach the ventricles. And then third is complete, a failure of all action potentials to reach the ventricles. So obviously, third is worse than second is worse than first. Lightheadedness, fainting, irregular heartbeat, and chest palpitations. Premature ventricular contractions result from stress, stimulants, or sleep deprivation. Abnormal action potential within AV nodes or ventricles, not detrimental unless they occur in large numbers. Your atrial fibrillation is chaotic timing of atrial action potentials. Ventricular fibrillation is chaotic electrical activity in ventricles. So fibrillation meaning chaotic timing, basically. Uncoordinated leads to death of heart cells, treated with a defibrillator. All right, so now looking at the cardiac cycle. This is all the events from the start of one heartbeat to the start of the next. So we've looked at the electrical events, we looked at the mechanical events, now we're gonna kind of put it together in a picture. The cardiac cycle includes both systole, which is contraction, and diastole, which is the relaxation. Contraction is when there's pumping blood. Diastole is when it's filling with blood. Referring to the chambers here. The contraction increases pressure, relaxation decreases it. Blood moves down its pressure gradient from high to low, and the valves ensure that the flow is forward, closure preventing backflow. The ventricular activity is the most important driving force. Your ventricular contraction raising your ventricular pressure, the AV valves push closed, and the semilunar valves pushed open as blood is ejected into the arteries. Again, this is both left and right ventricles going to the lungs and the body. Your ventricular relaxation lowers ventricular pressure. Your semilunar valves out of the heart are going to be closing, so because there's no pressure from the ventricles keeping them open. And the AV valves are going to start to open because there's no pressure from the ventricles closing them. So when your cardiac cycle begins, all four chambers are at rest. You can just assume that they're all at rest. AV valves are open, semilunar valves are closed. You have blood returning to both of your atria. So at the beginning of the cycle, the blood is coming into the right atria from the body, the left atria from the lungs. And this passive filling of ventricles, because the AV valves are open at this point, the ventricles are relaxed, the AV valves are open, and so you have this kind of passive blood is coming into the atria, and it's just kind of trickling down into the ventricles as well. And your atrial pressure is greater than your ventricular pressure. Semilunar valves closed, said that. The atrial contraction, the ventricular filling starts the cycle. The SA node starts the atrial excitation. The atria contract, pushing the remaining blood that didn't just passively go into the ventricles into the ventricles. And the ventricles are filled to end diastolic volume. So the end diastolic, meaning at the end of their resting phase, what is the volume in the ventricles? This is important because they can only pump what they have. So the EDV determines how much the ventricles can pump. After this point, the atria are relaxed for the remainder of the cardiac cycle until they get filled again. Between the atria contracting and the ventricles contracting, once they've filled, you have this also isovolumetric contraction that I mentioned. The Purkinje fibers initiate the ventricular excitation, and the ventricles contract, the pressure starts to rise, and the AV valves are pushed closed, but the ventricular pressure is still less than arterial trunk pressure, so the semilunar valves stay closed. So the pressure in the aortic trunk and in the pulmonary trunk is still greater than the pressure in the ventricles at this point. The ventricles are filled, but they haven't really started contracting fully yet. They've only kind of started contracting, just to kind of make sure essentially that the AV valves are closed. Then you have ventricular ejection. So after the isovolumetric contraction, you have ventricular ejection, which is the ventricles continue to contract so that ventricular pressure rises above arterial pressure. The semilunar valves are forced open as blood moves from the ventricles to the pulmonary and aortic trunks. Your stroke volume is the amount of blood ejected by each ventricle. So the stroke volume, the amount of, in each contraction, how much blood is your heart actually pumping. The ventricles have their EDV, but does EDV actually equal SV? The end systolic volume is the amount of blood remaining in the ventricle after contraction finishes. So essentially your ESV, so systolic meaning the contraction phase. So the end of the contraction phase, what's the volume? The ESV should equal the EDV, the end diastolic volume, what their volume was when they were filling, minus how much they pumped. It's not always going to be completely equal. After the ventricular ejection, you get the isovolumetric relaxation. This is when the ventricles relax, start to expand, lowering their pressure. The arterial pressure is greater than the ventricular pressure. By sliding back towards the ventricles, blood closes the semilunar valves. The AV valves remain closed at this point. They're not open yet because the ventricles are still starting to relax. So ventricular pressure is still a little. Ventricular pressure is still a little higher than your atrial pressure. And then when all valves are closed, blood neither enters nor leaves, and the time is called isovolumetric. So this is before the atria start passively filling the ventricles. Atrial relaxation and ventricular filling. All heart chambers relaxed. Atrial blood pressure forces AV valves open, and blood flows into your ventricles because there's, again, all chambers relaxed. The atria are filling from the lungs, from the vena cava. And the semilunar valves remain closed since arterial pressure is greater than ventricular. Here is just a depiction of it. This is very nice and healthy because it's actually helpful because it shows you where the blood is. It talks about the chambers that are working, the pressures that are occurring, and what the state of the valves are. You should be able to know all of this information. Yep. Okay, ventricular balance. Equal amounts of blood are pumped by left and right sides of the heart. The left heart pumps blood farther, and so it must be stronger or thicker. But ejected blood volumes must be the same or edema will occur. So what comes in has got to go out, basically. This is measured by cardiac output. The cardiac output is the amount of blood pumped by a single ventricle in 1 minute. See, I don't know if I can stand over on this side because I've got things organized. I'm going to have to hang out here. Amount of blood pumped by a single ventricle in 1 minute. I'm going to lean in. It's measured in liters per minute, and it's a measure of the effectiveness of the cardiovascular system. It increases in healthy individuals during exercise. That's why you hear athletes like, oh yeah, my resting heart rate's 45 beats per minute. Probably not. But they have a more effective heart, so it can pump fewer times and be just as effective. And still deliver adequate perfusion. It's determined by your heart rate, which is the beats per minute, and your stroke volume, which is the amount of blood ejected per beat. So basically it's rate times force. HR, heart rate, times stroke volume, SV, equals cardiac output. 75 beats a minute times 70 mils per beat is 5.25 liters per minute. That's our average cardiac output. You should know these numbers. So cardiac output must meet tissue needs. Individuals with smaller hearts have smaller stroke volume and so have to have a faster heart rate. Think about women and children, but also like mice. They have those little rabbits, little small mammals. They have really fast heart rates. Individuals with larger hearts have larger stroke volumes and slower heart rates. So endurance athletes have larger and stronger hearts typically. One of the ways that they will kind of evaluate racehorses is how big their nostrils are. The bigger their nostrils, the more air they'll be able to pull in. So the better, more fit they'll be. And who was it? Secretariat. One of the reasons why he was such an incredible horse, because even though he was smaller, his heart was huge, and that's actually what killed him, is that he had an enlarged heart. But his heart was so big, he was able to generate a lot more force, even though he wasn't, as big as some of the other thoroughbred horses. Your cardiac reserve. This is the capacity to increase cardiac output above rest level. This is basically like that extra bit of energy that you can always come up with. So that you have your baseline that you can use, and then if you need to do more, if you need to be able to do exercise, your heart has this capacity to be able to move beyond just your regular 75 beats a minute. 5.7 liters, 5.25 liters per minute output. It can do more. You subtract cardiac output at rest from output with exercise, and you get the cardiac reserve. Pretty straightforward. I'm not going to make you do a ton of math, by the way. Don't worry about that. Your heart rate accelerates and stroke volume increases during exercise. This gives measure of level of exercise an individual can pursue. So your cardiac output can increase 4-fold in healthy non-athletes and up to 7-fold in an athlete. This is why a lot of times if you haven't been exercising a lot before and you have some other kind of health issues, it's always important to ask your doctor to make sure first, because you may be able to increase your cardiac reserve up to 4-fold, but your cardiac reserve might not be that much. Maybe you only have a 2-fold cardiac reserve, and so they do stress tests to help determine that. All right, variables that influence heart rate. Your chronotropic agents change the heart rate. So chrono, time. They alter the activity of nodal cells, your SA and your AV node. So you need to know which feature of the cardiac cycle they affect. So they're changing the heart rate. And you need to know what they're impacting. They're impacting the nodal cells. They often work via the autonomic nervous system or hormones, so they can do either one. So this is not just one thing. These are agents. These are multiple different things, chemicals or messengers or signals that stimuli that can affect your heart rate. Positive chronotropic agents increase your heart rate. That would be sympathetic nervous stimulation from norepinephrine, either from the adrenal glands. The epi and the norepi bind to nodal cells and increase their firing rate. The receptors on these cells are beta-1 adrenergic receptors. They go through the adenylate cyclase G-protein cascade. We learned that in chapter 17. And then the phosphorylated calcium channels enhance calcium influx, so cells fire sooner. That's going to help to depolarize the cells. So this isn't about contracting. This is about force of contraction. This is about rate of contraction at this point. There is just a depiction of that. It's just this norepinephrine being released, and then that G-protein cascade. Thyroid hormone increases the number of beta-1 adrenergic receptors on the nodal cells, so that will increase the binding of norepi and epi. Caffeine inhibits the breakdown of cyclic AMP, which will allow for more kinase activity. Nicotine increases the release of norepinephrine, and cocaine inhibits the uptake of norepinephrine, meaning that more stays in the cleft longer to continue stimulating the heart cells. So all of these are going to increase your heart rate. If you can't remember the rest of them, just kind of group them all together, and you should be able to remember that caffeine, nicotine, and cocaine are all going to jack you up. Negative chronotropic agents decrease the heart rate. This comes from parasympathetic activity, axons releasing acetylcholine. Acetylcholine binds muscarinic receptors, which are potassium channels. That opens and exits the cell, making it more negative, so it has a longer time for the nodal cells to reach threshold. Beta blocker drugs, they're going to interfere with epi and norepi binding to the beta receptors to treat high blood pressure. Your autonomic reflexes are also going to influence your heart rate. Your baroreceptors and chemoreceptors send signal to the cardiac center and your medulla oblongata. That's going to influence sympathetic, parasympathetic systems to alter the cardiac output. Your atrial reflex, also called your Bainbridge reflex. This is probably named after some dude that I don't want you to have to remember, so just remember atrial reflex, because again, this tells you nothing specific. He just wanted to name it after himself. The atrial reflex protects the heart from overfilling. It is done by baroreceptors in the atrial walls, stimulated by increased venous return. So increased return of blood from your veins. The veins are reservoirs of our body. So once blood starts to come back from there, if you've been resting for a long time, if you've been doing an international flight, you'll have all of this return to your heart. And the baroreceptors, the stretch receptors in the atrial wall will recognize that extra stretch. You have increased nerve signals to the cardioaccellatory center, increasing the excitation of sympathetic axons to the heart, which increases heart rate to move blood through quickly, and your atria stretches less. So it increases your heart rate. So instead of kind of saying, okay, we'll just go slow and let the heart really, really stretch and pump a bunch of blood at once, which could be very damaging, it just tries to pump faster, so that way there's not as much blood sitting in the atria as long. So that's your atrial reflex. Stroke volume is the amount of blood ejected in one beat. So now we're going to talk about that variables that influence stroke volume. Stroke volume is influenced by your venous returns, the amount of blood that's coming back into your right atria, by your inotropic agents, these alter the force of contraction rather than chronotropic, and the afterload, which we'll see is the force that the heart has to work against in the atria, and the aorta, and the pulmonary arteries, or pulmonary trunk. Your venous return is the volume of blood returned to the heart, directly related to stroke volume. However much you're going to pump out starts with how much you have. Determines the amount of ventricular blood prior to contraction. That's your EDV, so you're in diastolic before your systole. And the volume determines preload. The preload is the pressure stretching the heart wall before shortening. Remember that cardiac muscle cells, they have to be filled to a certain point to be stretched and get to their optimal length before they can contract. If they're not filled enough, they won't contract fully. The venous return, you have your Frank-Starling law. Unfortunately, we got to know their names because we don't have another name for this law yet. Get your PhD and name it something else that's more specific than Frank-Starling law. As the end diastolic volume increases, the greater stretch of the heart wall results in more optimal overlap of thick and thin filaments. So that's basically what I was saying, that as your ventricles fill, they'll start to stretch the heart wall, the cardiac muscle cells to a certain point. They get that optimal overlap. The heart contracts more forcefully when filled with more blood, so the stroke volume increases. So if you go a little beyond your standard end diastolic volume, if you have a little more increased venous return, you're going to have an increased contraction. Venous return may be increased by venous pressure or increased time to fill. Venous pressure increases during exercise, and time to fill increases with the slower heart rate in high caliber athletes with strong hearts. It decreases with low blood volume, hemorrhaging, or a high heart rate. If your heart is pumping faster, you're going to have less returning at one time. It results in smaller end diastolic volume and a smaller preload, which results in a smaller stroke volume. Balanced ventricular output is due largely to the Frank-Starling law, saying that as one side receives increased blood, it contracts more forcefully, leads to greater fill and more forceful contraction on the other side. So balanced ventricular output, what comes in needs to go out. And this Frank-Starling law is basically going to say, okay, however much comes in, that will determine the stretch, and the stretch will determine how much is contracted. The inotropic agents, as I said, change the stroke volume, not the rate of stroke. They alter the contractility, the force of contraction, generally due to changes in calcium availability in the sarcoplasm, and the cytoplasm of the muscle cells. The calcium levels directly relate to the number of cross bridges formed, so the more calcium you have, the more you can form those myosin-actin cross bridges. Positive inotropic agents increase available calcium, epi and norepi via beta-adrenergic receptors, thyroid hormone, increasing number of receptors, and the certain drugs boosting cardiac output. Negative agents decrease calcium availability and lower contractility. If you don't have as much calcium, you can't form as many cross bridges. Electrolyte imbalances, such as increased potassium or hydrogen, and certain drugs, calcium channel blocking drugs, blood pressure drugs. Okay, so afterload, that is another thing that affects stroke volume. Your afterload is the resistance in arteries to the ejection of blood by ventricles. So the resistance in arteries, both in the aorta and in the pulmonary arteries, to the ejection of blood by ventricles. The ventricles want to pump blood from their chambers out into the arteries, but there's already blood in there. So they're going to be forcing against it. Remember, they're pushing up against those semilunar valves, keeping them closed until the ventricular pressure overcomes it. The ventricular pressure has to overcome afterload. That's the pressure it's overcoming. Right there. Atherosclerosis, which is plaque in the vessel lining, it's the hardening of the vessels, increases your afterload. You see it with aging, typically because as we age, we have just been gathering more and more bad habits. Your smaller arterial lumen exerts greater resistance to movement of blood. So the smaller the artery, the smaller the vessel diameter, the harder it is to push through. Think about blowing through a straw versus blowing through like a paper towel tube. It's a lot easier to blow through something that has a wider diameter because there's less resistance. It's a larger space. So the smaller lumen exerts greater resistance, resulting in decreased stroke volume. So the higher your afterload, the lower your stroke volume. Relationship of EDV, ESV, and stroke volume. Again, for the people that just like depictions, you can see your venous return, the inotropic agents affecting contractility, and the afterload, that pressure resisting. I'm not going to go through all of this because I already have. And yeah. So cardiac output varies directly with heart rate and stroke volume. Oh, did I? Oh, no, okay, we're good. Heart rate depends, so cardiac output varies directly with the heart rate and stroke volume. CO equals HR times SV, varying directly with it. Heart rate depends on chronotropic agents, influence the SA node to change its firing rate, influence the AV node to alter the amount of delay. The stroke volume, depending on the state of the myocardium, venous return altering the stretch, inotropic agents influencing cross bridges, and afterload as well, decreasing stroke volume. Here is a really wonderful little chart. I would recommend you making your own little version of this to help you understand what factors affect cardiac output. Chronotropic agents, venous return, inotropic agents, and afterload. All four of these are going to influence cardiac output in different ways. Your chronotropic agents are going to influence your heart rate. Your venous return, your inotropic agents, and your afterload are going to influence your stroke volume. Okay? You can see it just gives you just good information. Inotropic agents, they're altering the calcium levels. Positive agents increase stroke volume. Negative agents decrease stroke volume because they decrease the amount of calcium. Venous return, more venous return, so directly correlated with. So that means that the more venous return you have, the higher your stroke volume. The higher the afterload, the lower the stroke volume, inversely correlated. Same thing here, chronotropic agents, SA and AV node activity, positive agents increase it, negative agents decrease it. All right, bradycardia, tachycardia, we're almost done. Persistently low resting heart rates, below 60 beats a minute, normal change in athletes as their hearts get stronger, abnormal due to hypothyroidism, electrolyte imbalance, and congestive heart failure. Tachycardia is persistently high resting heart rate, over 100 beats per minute, caused by heart disease, fever, and anxiety. I have a friend who has POTS. She was diagnosed at 24, was a swimmer all her life, very, very active, but then she immediately, like over a summer, started developing this fainting disorder. And was eventually diagnosed as POTS. And essentially what happens is that her heart rate will go from being perfectly normal to 45 beats per minute. It'll just bottom right out. And she has, a couple of seconds to, she usually gets, you learn the symptoms basically. And so she kind of knows how to brace herself. And it's one of those very hard to diagnose, very hard to treat disorders because it affects you systemically. But it's, her heart rate dropped real low. It's scary. Development of the heart. The heart development begins in the third week with the formation of two heart tubes forming from the mesoderm and the embryo. The heart tubes fuse, forming a single primitive heart tube. It begins to beat by day 22. That's what I want you to know. That's the important part for me. It begins to beat by day 22. That's not so much. The sinus venous and primitive atria from parts of the left and right atria. Yeah, I don't get too big into heart development stuff. Let's see. Yeah, pretty pictures, nice folds, cool. Weeks 5 to 8, you get this single heart partitioned into 4 chambers, and your great vessel starting to form. A common atrium subdivided into left and right atria, divided by their interatrial septum. The opening in the septum is that foramen ovale. Your foramen ovale, so this is the part that you know. So really, when we're talking about development, what I want you to know is begins beating by day 22. Or sorry, rather, it begins the third week. So Day 22 is about 3 weeks in a day. So it begins in the third week, and you usually get beating done by day 22. And then I want you to know this about the foramen ovale. So the embryonic lungs are not functional. Blood gets shunted from the right atrium to the left atrium through the foramen. Because again, the blood coming to the fetal heart is already oxygenated. This pushes the septum primum into the left atrium, and at birth, the left atrium pushes the septum primum and secundum together and closes that septum to form the fossa ovalis, which is the remnant of that opening between the two septum, between the left and right sides, between the left and right atria. The interventricular septum divides the left and right ventricles, grows up from the floor of the ventricles. Atrial septal defects. A postnatal heart still with opening between the left and right atria. So blood is coming from the left atrium, is shunted into the right atrium, lowering its pressure and can lead into enlargement of the side of the right heart, or the right side of the heart. Sometimes this will close up on its own. Sometimes they have to go in and actually perform surgery to close it up. But essentially what you're having is this mixing of blood from the left side coming back into the right side. So you have this oxygenated blood that's going, stretching further the right side of the chamber. Your ventricular septal defect occurs if the interventricular septum is incompletely formed, and the tetralogy of Fallot is an aorticopulmonary septum divides the truncus arteriosus unevenly, resulting in ventricular septal defects, narrow pulmonary trunks, aorta overlapping ventricle, and enlargement of the right ventricle. And that's the whole chapter.